Hey everyone, welcome back. Bio 232. We are discussing blood, and we're about to do erythrocyte disorders, and then we're going to move into white blood cells as well. So let's go ahead and get started with the lecture notes. Erythrocyte disorders are listed in your book on pages 650, 651. So you can read about them there. We are going to highlight a few things about them. We're not going to go into a lot of detail, but a little bit of detail. So let's do that now. So the first thing is anemia. We want to know what that term is. We've heard it. I'm sure you've heard of anemia before. And this is a condition that lowers the oxygen carrying capacity of the blood. Now, this can be due to numbers of things, right? Like if you don't have enough red blood cells, then you don't have enough hemoglobin in those red blood cells to grab onto oxygen and carry it around. Or you could have plenty numbers of red blood cells, but inside of them have low hemoglobin. So there can be different ways in which anemia can occur. So the first one there, this one, is insufficient numbers of red blood cells. Okay, so that's the number one. Let's put a number one here because it's there's two different ways. So we'll put a number one right there, and we'll put a number two right here. And then there's a third way, which we didn't mention here, is not only can you have low hemoglobin numbers, but you can have hemoglobin that just doesn't work correctly. So you can have the right numbers of hemoglobin, but you can have it not be working properly. So we'll get to that in a second. So those are the three main ways. So first, insufficient numbers of red blood cells, okay? And this is going to mean that you're going to have low hemoglobin, because if you don't have enough red blood cells, there's not enough hemoglobin. Red blood cells are big bags of hemoglobin. So there's a couple different anemia types. And so really what you want to think about is, what is the name in front of the anemia saying? Hemorrhagic anemia. Well, a hemorrhage means that we're losing blood. Okay, this is supposed to be an up arrow here. Let's make that an up arrow. Sorry about that. Up arrow. Okay, there we go. Due to increased blood loss. So we want to think of things like a severe wound, hemorrhoids, ulcers, extreme menstrual bleeding. SS stands for signs and symptoms, and so the most common symptom is fatigue. Hemolytic anemias. Now, lytic makes us think of the word lyse, and lyse means destroyed, and that's what's happening here. We're having destroyed cells, red blood cells, lyse prematurely due to lots of different things. It could be due to hemoglobin abnormalities. We're going to talk about those in a minute. Bacterias that are in the blood that are destroying the red blood cells, or other infection. Okay. So this can be a problem. We're going to talk a little later about Rh negative, Rh positive. This is a situation where mom can sometimes attack the fetus's blood because it's different than hers, and will destroy the blood, and then hemolytic anemia will occur in the fetal blood. So we'll come back to that in a future lecture. Then we have aplastic anemias, and this is due to the impaired function or impaired formation or destruction of red bone marrow, because if our red bone marrow is not working, then we can't produce the right amount of red blood cells. So this is usually due to drugs, toxic chemicals, radiation. And one of those drugs or toxic chemicals, for example, can be chemotherapy. So even though chemotherapy is useful because it helps us try to not to destroy cancer cells, right? Unfortunately, it will destroy other fast-growing cells, that rapidly dividing cells, so it can destroy the red bone marrow cells, the stem cells there. Okay, so and because it can destroy the stem cells, all three blood types can be affected, meaning white blood cells, red blood cells, and platelets. Now, decreased hemoglobin content is what we were talking about up here when we said low hemoglobin levels. We're not getting enough hemoglobin into the red blood cells for some reason. So one is iron deficiency. Okay. Another one is vitamin B12 or intrinsic factor deficiency. All right. And the reason of that is vitamin B12 is important. Well, here, let me go back. So the iron deficiency usually is due to we don't have enough iron intake. However, we can also have low levels of B12 or folic acid, right? And the reason why is that these are important for DNA synthesis, and we need to have DNA synthesis for red blood cell production. DNA synthesis, and therefore affect RBC production. Okay, so if we don't have enough iron, we can't make hemoglobin. Remember, there's an iron group in the middle. If we don't have B12 or folic acid, we can also not make the globin portion of hemoglobin. So as a result, what we'll get in this case is we'll make the red blood cells, but they will not have a lot in them, so they'll be small. They're called microcytic anemia. Now, we can also have low level of vitamin B12 or intrinsic factor, which causes another type of anemia, which is macrocytic anemia. And in macrocytic anemia, red blood cells grow, but they don't divide. And we get these large pale cells where we might have enough iron, but we can't use that iron to make hemoglobin because the vitamin B12, or the thing that allows vitamin B12 to be absorbed, isn't there, and so we can't make the globin portion. So we get big cells, red blood cells, that are very pale and not very full of hemoglobin. But in either case, we have not enough hemoglobin, and therefore we're going to be anemic. Now, the third one is abnormal hemoglobin. And abnormal hemoglobin usually is derived from genetic states, genetic conditions, and for reasons due to the way the proteins are made, the globin portions are unstable. So there are two types. There's thalassemias and sickle cell anemia that we'll discuss. There's others, but in thalassemias, the red blood cells are thin and delicate, and the globin chain is not properly formed. And in sickle cell anemia, there's an abnormal hemoglobin called hemoglobin S. And what happens, and I'll show you real quickly in the book, because this is on page 651, figure 17.8. If you look down at the bottom of this diagram here, there is a single change in an amino acid that makes the hemoglobin in individuals who have sickle cell disease. So this is supposed to be glutamic acid or glutamate, number 6, and instead it's valine. And that one change will change the shape of the red blood cell into this, especially under stressful or low oxygen level situations. And it gets a sickle shape, and then the red blood cells start sticking together and clogging up little capillaries and not allowing oxygen to get to its location. So it says crescent-shaped cells rupture during increased demand for oxygen. Okay, so when we have low oxygen levels. So anyway, those both, because they do not make the globin portion correct, we won't get a working hemoglobin, and therefore oxygen transport will be less. All right. Now, to move to, so those were all low red blood cells. Now we're going to get to polycythemia. And polycythemia is the opposite. It's an increased number in red blood cells, causing sluggish movement of blood. Polycythemia vera is one type It's often due to like bone marrow cancer, where the cancer is in the bone marrow and it's rapidly dividing and it's making lots of cells, and as a result, your red blood cell count might go up to from 5 to 6 to 8 to 11 million red blood cells. So as you can imagine, the blood volume is going to get really thick. And if our blood volume, or the viscosity of our blood gets really thick, that might cause increased blood pressure or an increased chance of clotting because the blood is so thick and sluggish. Secondary polycythemia is usually due to something else and it's temporary, so it's usually due to low availability of oxygen, like high altitudes or chronic anxiety, something like that. And what'll happen is, in this case, you will because there's not a lot of oxygen in the environment, or you're anxious and you're not breathing correctly and you're not getting enough oxygen into your system, your body starts to make more red blood cells in hopes that it can grab onto more oxygen. And so this is what happens if you go to high altitude. The first couple days you may not feel very well because you're not getting enough oxygen in, and then you will start to make more red blood cells. And after 3 or 4 days, as the number of blood cells increase, and you can then grab onto more of the oxygen in the air, even though it's very thin, you'll start to feel better. There's a thing here about blood doping too, which you can read, but I'm going to not cover right now. But is interesting. And let's move on now to white blood cells. So white blood cells have a nucleus. They are not like red blood cells where they have ejected their nucleus. They don't contain hemoglobin, obviously. That's supposed to be HGB, not HBG, excuse me. And these formed elements defend the body. That's their function, right? They use either inflammatory or immune responses against foreign invaders like bacteria, viruses, parasites. And they also will work to help clean up and remove debris from injured areas of the body. There's a lot less white blood cells per millimeter cubed than red blood cells, as you can see the difference there. And it's good to know these two terms. I think you have to know them also for lab this week. So leukocytosis is more than about 11,000 white blood cells per millimeter cubed, and leukopenia is less than 5,000. So that in between 5 and 11 is our normal number, and if you go a little above or a little below, you may be having some problems or issues. If you have more, that often means you have some sort of infection because you're mobilizing and making more white blood cells. If you have too few, sometimes you have some other disease or disorder which is repressing the creating of or destroying the white blood cells. Now, white blood cells primarily act in and on tissue, so they're in the blood, but they leave blood circulation. They leave blood circulation by diapedesis, and this just means that they exit out. They go out through the capillary wall. So let's type that in there. They go out through the capillary wall. We'll talk in a future lecture about how capillaries have these little tiny pores, and the white blood cells can go through those. And they find where to go by the process of positive chemotaxis. And in positive chemotaxis, they are in effect following chemical substances that are released by the affected tissue and other damaged white blood cells. And so they're kind of like bloodhounds. Bloodhound picks up the scent of something and then follows the molecules in the air of that scent, and they get stronger. The bloodhound nose is getting closer and closer to whatever it's trying to find, and the white blood cells do the exact same thing. They kind of notice there's a chemical somewhere, they orient in that direction. If they get more chemicals, they orient towards that, and they eventually find the affected tissue. Now, leukopoiesis, remember, erythropoiesis is white blood cell production. Leukopoiesis is erythropoiesis is red blood cell production. Leukopoiesis is white blood cell production. If we say hemopoiesis, we're referring to any blood cells. And there are definitely hormones that are involved in this process, and those are called colony stimulating factors. They're stimulating colonies of white blood cells. or interleukins. And interleukins are signaling molecules which will release and due to immune system problems or injuries, and the white blood cells will react to them. Okay, these same factors also increase the potency of mature leukocytes, meaning that when these two kinds of chemicals are released, they also make the mature leukocytes that are already there more active. They kind of pump them up a little bit. So leukocytes fall into two major groups, and these are going to follow very similar to what you're going to be doing in lab this week, looking at some of the different blood cells. We have granulocytes and agranulocytes, and all of them do immune response. The granulocytes, right there, are also called polymorphonuclear leukocytes. Polymorphonuclear. Let's break that word down. Nuclear means nucleus. Poly means multiple, morpho means shape. And so what we're saying is, instead of having a nice round nucleus like usual, look at this nucleus over here. It's got these multiple lobes. See those three different lobes there? And so it's got poly, multiple, morphed or changed nuclear shapes. And so we call those different lobes. So the first one, so there's so if you look here, granulocytes, and then we've got neutrophils, eosinophils on the next page, and basophils. We have those three. And then for agranulocytes, which is the second category, there's lymphocytes and a couple different kinds, and then on the next page, monocytes. So there are 2 agranulocytes and 3 granulocytes. Now, they're called the granulocyte, because they have cytoplasmic granules. They have these little dots that you can see in the cytoplasm, and they have that lobed nuclei. Okay, that's the easy way to figure out what they are. Functionally, they are phagocytic. They will go around and engulf things. So neutrophils, let's start with neutrophils. So neutrophils are the most numerous. You're going to see those when we're looking at the blood slides more than any others. The nuclei have 3 to 6 lobes, and the cytoplasm stains a pale red. Very fine granules are there. Okay, we always see granules. And these granules have lysozymes. And lysozymes are things that will destroy bacteria, and also there's others called defensins as well. So both these lysozymes and defensins are molecules that will destroy and break down bacteria, and they will phagocytize them. Okay. And note it says an increase in neutrophils may indicate bacterial infection, and that makes sense because these guys are going to kill bacteria. So if you see a lot of them, you might have a lot of bacteria on board and your body's trying to destroy them. All right. Next, we get to eosinophils. And eosinophils are often in areas kind of on the kind of near membranes, and they're protecting us against, they are protecting us against parasitic worms, protozoans that are in like water, untreated water, for example. And they also have some allergy-related responses because they are in these areas that are near the skin and mucosal membranes. As many of them do, they'll leave the capillaries and enter tissues. They produce the enzyme MBP, which stands for major basic protein, and this is what attacks the worms. This is what attacks the parasitic worms. Attacks the worms. And then antihistamines, and that's where the allergy response is coming from. They are phagocytic, like we said, but they also ingest antibody-antigen complex rather than just bacteria and virus. And what that means is if there's a foreign invader floating around and you have an antibody that can attach to it to kind of flag it and also try to make it inactive, eosinophils will see that this has happened and then go around and engulf those. So it kind of works with other immune systems in your body to reduce foreign invader count. And we'll talk more about that later when we get to lymphatic system. So for now, don't worry about it. But antibodies flag, I mean, I guess what we can write in right now is that antibodies flag foreign invaders. Okay, and so they're basically attaching themselves to foreign invaders to slow them down, to make them inactive, and to tell things like eosinophils, hey, why don't you engulf this thing? Okay, we'll come back to that in a few weeks. Basophils, so these are the rarest ones. These are the hardest ones you're going to see on your slides. Function's not exactly known, but it appears that they kind of act like mast cells in the connective tissue. We talked about those in 231. They do contain histamine and heparin in their granules and maybe even some serotonin. And so they basically have allergy responses. Allergy, oops, if I can spell it right, allergy responses, right? Allergic or allergy responses. Okay. So those are the three granule sites. And if you looked at every picture, you always saw granules. Pink granules appear in the eosinophils, these really dark, big granules in the basophils that hide the nucleus a little. Now we get to agranule sites. These don't have granules in them. Lymphocytes are broken down, are often found in lymphoid tissue, our lymph system. And there's two main types, the T and B lymphocytes, okay? Notice how they look. There's a gigantic nucleus and there's very little cytoplasm. You can see a little sliver of cytoplasm there. There are no granules in it. Notice how smooth it is. One way you tell the difference between these in lab is this has got this smooth nucleus with no granules and a little sliver of cytoplasm. And here, the nucleus is obscured by all of these granules and it has kind of a bumpy appearance. So that, if you look at them quickly, they kind of look similar, but if you really look at them, they don't. So in T lymphocytes, what do T lymphocytes do? You've probably heard of these before. They attack foreign cells. And they also release chemicals to regulate the immune response. So what they're really going for is any foreign invader, like viruses, tumor cells, cancer cells, fungi, are directly attacked and destroyed. So these are sometimes called killer T lymphocytes because they go right in and they destroy things. And they have what we call a cell-mediated response. And this is because they directly destroy them. The cell does the work, directly destroy. Now, B lymphocytes are involved in antibody-mediated immunity. And remember up here we said antibodies flag foreign invaders. Okay? So what B lymphocytes do is they will become plasma cells, and plasma cells produce antibodies. And as we said before, antibodies are special proteins that inactivate antigens. They stick to an antigen. Now, antigens are foreign substances to us. That we don't recognize as part of our body. And so when we see something in our body that's foreign to us, we want to destroy it. We don't want it to be there because we think it could harm us, okay? And so this antigen antibody response helps combat infections, either viral or bacterial, and it helps us lend immunity to some diseases. So when you get a flu shot, you're getting an antibody, a broken up piece of the flu virus, and you make which is an antigen, and you make an antibody towards it using these plasma cells. It takes a few days to a week to make antibodies. And then if you were ever to get that actual flu virus in your body, your antibodies that you made would still be there. They're still being made in low numbers, and they would attach themselves to that virus. So that's a very useful long-term immune response that we have. And antibodies are also responsible for blood typing. We'll talk about blood typing in a future talk. All right, now our very last white blood cell that we need to discuss are monocytes. These are usually the easiest ones to see on the on slides because they are the largest ones, and their job is kind of to be like a cleanup crew. They become macrophages. And what they do is they go around and they eat cellular debris. Okay, they will. So if you have a viral or a bacterial infection, tissue's getting damaged by those cells, and so the monocytes will go around and kind of eat and destroy all the different things that are there. and kind of clean up. So we see these a lot, increase in number of chronic infections. All right, so in viral and bacterial chronic infections, we see a lot of monocytes usually. Now, interestingly, these cells are produced in a little different places. So the granulocytes are produced in myeloid tissue, which is the red bone marrow tissue. Agranulocytes are produced there, but they're also produced in lymphoid tissues. And so this is why we call B and T cells lymphocytes, because they can be made in the lymph tissue. So that's good to remember. All right, we're going to do one more little bit here and then finish up this part of the lecture. We're just going to talk about thrombocytes. So thrombocytes are platelets, right? The same name, mean the same thing. And they come from megakaryocytes. So now if you remember when we talked about red blood cells, and white blood cells, they all come from a hemocytoblast. And then they go down different developmental pathways. And so for this one, all we want to really know is that the stem cell is the hemocytoblast, and it will go and eventually become a megakaryocyte. What the megakaryocyte is what it does is little fragments get broken off of the megakaryocyte. So it's a large multinucleated cell, and it sheds fragment of its cytoplasm. Not complete cells, but little fragments. And you can see these little fragments right there. Okay? And so each platelet or each fragment is has part of the cell membrane, and there's stuff inside. Okay? And the stuff that's inside, the chemicals that are inside, are going to all these chemicals here are going to help us with the clotting and blood and hemostasis, which we'll talk about in our next lecture. So some of these terms that you're seeing right here that we haven't talked about yet, we're going to talk about in the next lecture. Okay, they have about a 5 to 10 day lifespan, and they prevent blood loss by initiating the clotting mechanism and causing a chain reaction. So that's their function. Okay, so their function is that. I will tell you the name of so ADP. You know ATP, and ADP is spent ATP, right? We've removed a phosphate to make some release some energy, and PDGF is platelet derived growth factor. Okay, so a growth factor that comes from platelets. Okay, so we're going to talk about that in the next lecture, hemostasis, and we're going to do a whole lecture on hemostasis, and so that one will be where we head next. Okay, so hopefully that was a good overview. There's a lot of information in there that's useful for lab as well, because you're going to be looking at red blood cells, white blood cells, and platelets in lab, and so it's good to see all those different structures and talk about them a little bit and their functions. All right, and so we'll see you at the next lecture.